Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Dr. Marom Bikson, and he is a biomedical engineer at City College at the City University of New York City. And he's been there for a long time, but 11 years. And he focuses mainly on TDCS, Transcranial Direct Current Stimulation. And basically what that is putting electrodes on your head, and that changes the sensitivity essentially of the neurons. And then you can do different things, learn faster, or have different anxiety reductions or whatever. And so he focuses on this. He also has spun off a company called Soterix from this technology as well. He runs NYC Neuromod, which is a big conference focusing especially on the New York side of things. And then he does consulting as well. So we talk about all these things, really somebody that does a lot of different things, got his fingers in a lot of dikes, and I found this very fascinating. So, enjoy. So, Dr. Marom Bikson, pleasure to have you on the show. You are a professor at City College of the City University of New York City, and we were just having a laugh at this a little bit before, and you also have a company, and you work on transcranial direct current stimulation, and do lots of consulting. So I think this is going to be a really fun talk. Do you want to introduce yourself a little bit and kind of what your specialty is?
Yeah. Sorry, do I need to hit the record button on my end or no?
No, it's already being recorded.
Sorry. Okay. So my name is Marom Bikson. I'm a professor of Biomedical Engineering at the City College of New York. My lab does a lot of work on neurotechnology, from basic science to product translation. I also have a company I co-founded called Soterix Medical, which is a neuromodulation company.
Very cool. And so you also... So you founded Co-Direct Neural Engineering Group, and there, I want to talk about this. Is really interesting article on there, new treatment for neuro COVID. Do you want to talk about this a little bit?
Yes. So, obviously the pandemic we're going through needs no introduction. And I think it's also become very aware, people, the public in general has become very aware that there are many people who, after overcoming sort of the initial assault of the virus, have lasting symptoms. They don't go away. Some of those lasting symptoms seem to be neurological or psychiatric in origin. So people who have never had headaches all of a sudden have headaches. People suddenly have the ability or difficulty focusing to the point that it impairs their ability to work. They're constantly fatigued. So this all falls into this sort of category of things that people are calling neuro COVID. And it's almost like another wave, like a second epidemic that's going to be hitting us. And so I'm very motivated to see whether any of the technology that I've been working on or that my colleagues have been working on can be used to address this neuro COVID, these long COVID neurological or psychiatric issues.
Okay. And so how do you do that?
Well, we already have some tools at our disposal, right? We have technology called Transcranial Direct Current Stimulation, or TDCS, that I and others have been developing now for several decades. There's technologies to stimulate the vagus nerve. I know that's something that you've covered on your podcast as well. You can stimulate this is, by the way, non-invasive techniques. You can stimulate the vagus nerve non-invasively via the neck or via the ear. There's a branch of the vagus nerve there. And so in general, what we're looking at neuronal targets, pieces of the brain, of the nervous system, that we can apply energy to in order to activate them in order to trigger what we hope are therapeutic processes for neuro COVID. And in many cases, in many cases, there's already work, for example, on migraine. And so now that you have this emergent form of migraine, part of this neuro COVID, maybe it's not so much of a leap to say, well, maybe it will work for these people's migraine. There's a lot of work on looking at depression and mood. So again, when people develop mood disorders, once again, we can say, well, we already have a tool that we're testing for that purpose. Let's simply translate it and reapply it for this new population.
So what's kind of the mechanism behind that? How does it work? I mean, you kind of stimulate the vagus nerve, or do TDCS kind of have a calming effect? But is it something to do with a calming effect, or what?
So I think the mechanism will depend on the modality. And I think I see different neuromodulation approaches as different as two different drugs. Like that's the level of there could be engaging fundamentally different mechanisms in the body, so they're leading to fundamentally different outcomes, and that's a great thing. So when we're talking about transcranial direct current stimulation, transcranial means through the cranium, and the thing that you're stimulating is targets inside the cortex. So you're trying to identify targets centrally that you think that you can activate, to lead to different therapeutic outcomes. When you're stimulating outside the skull, you're stimulating cranial nerves, like the vagus nerve. You could also be stimulating peripheral nerves. So now that's a different target. It's still leading into the nervous system, but obviously through a fundamental different way. So I think we have these different tools in our arsenal, and we can always try to pick the right tool for a given indication. Like, for example, with mood, there's a region of the brain called the dorsolateral prefrontal cortex that is believed to be involved in mood regulation. FDA-cleared technologies like transcranial magnetic stimulation already stimulate it. We're also targeting that region with TDCS. So that would be an example of using TDCS to try to improve mood.
Okay. And what are some of the results of this so far?
So I should say some of these technologies are investigational in the US, so they may have been approved abroad, but in the US, the majority of things I'm going to be talking to you about are investigational. So I want to say that up front. However, there have been some very encouraging clinical trials. These are randomized controlled trials. This includes TDCS for depression, where there's been some very important work published, including in the New England Journal of Medicine, showing significant improvement in mood. There's been a lot of work on TDCS. Again, I'm going back to TDCS, showing that it can have improvements in people suffering from neuropathic pain, chronic pain, migraine, fibromyalgia. And one important note is, in many cases, these are the patients who are the sort of the hardest to treat of the hard to treat. The fact that they are enrolling in these trials sort of implies that they've already failed, right? If there's something good enough already out there, whether it's, behavioral therapy, wearing sunglasses, taking a conventional pill, if that had worked, they probably would not be enrolling in these trials. So these are already very difficult, drug-resistant forms of the disease, where we're seeing very positive signal that TDCS can be effective. And I mean, you can extend that story to neuromodulation in general.
Very cool. So if people think they have the COVID brain, do they just strap up some electrodes to their head and self-administer, or what are some potential risks involved with this?
One of the big motivations to get FDA clearance has to do with making this accessible, right? As long as it's not FDA cleared for a given indication, that does present a hurdle for people accessing the treatment. And so, as I mentioned right now, TDCS in general is not FDA cleared for any medical use in the US. So in the case of the effort I'm talking about right now with neuro-COVID, these are part of clinical trials. So patients would have to enroll in these clinical trials. They would have to meet inclusion criteria. And obviously that is a limited sample. There is also the possibility that they may be able to access these treatments off-label. So obviously it's possible for physicians, for different medical centers to provide treatments that they think are in the patient's interest, not precisely on an FDA label. And so you have centers like NYU, where their neurology program has a very robust telemedicine-based program. TDCS is part of that telemedicine-based program. And so that's another example of a different kind of avenue where patients may be able to get access to these treatments off label, but under medical supervision.
Okay. So still a little bit experimental, but if you do feel especially bad, then if you are kind of in this worst of the worst category where nothing else has helped, then this could be a good potential solution.
Yeah. But let me just say about that. I know you're right, but about that term experimental, let's say TDCS is approved for PTSD on Thursday. Okay, so on Wednesday it was not approved, and on Friday it is, but it's the same thing. So on Wednesday it was experimental in the sense that it has no FDA clearance, and on Friday it is. So I perfectly agree with what you're saying, and we have to be very careful how we talk about things when we're talking about off-label. But I also, it's not necessarily the case that something that is experimental would necessarily work less well. And it's also the case that obviously very different people respond to different things in very different ways. So something may be experimental but work for person A, and something may be FDA approved but not work at all for person B.
Mm-hmm. Yeah, no, that's a very good distinction, and yeah, it's all just kind of labels that we put on things. So do you want to explain? Literally.
Yeah.
Do you want to explain TDCS, how it works, why it works, all this kind of stuff?
Yeah, I love to talk about that. I've been thinking about that for my PhD was about how direct current stimulation affects neurons. The big idea in principle, it's simple. Obviously, the brain is not simple. So there's a lot of layers to this onion. But in principle, it's very straightforward. TDCS works by the application of direct electrical current to electrodes placed on the scalp. The current flows, there's always at least two electrodes. The current flows from one electrode to the other electrode. People will use the term anode and cathode to describe these two electrodes. The current is flowing through the head as if the head is a light bulb. And in the center of that light bulb is a brain. So as that current flows through the head, it will wash over the brain. So the neurons inside your brain are being washed in this direct electrical current. This is something that we know and we've studied extensively. When a neuron is washed in electrical current, it responds to that electrical current. And the reason is that neurons are electrical organs. Each one is like a little mini electricity generator, generating action potentials. It has a resting membrane potential. They're little electrical organs. And when you wash them in current, they polarize and their function changes. Now, how they change is going to be a function of the nature of that electrical current. If that electrical current is DC, and if it's low intensity, they will change in a certain way. If it's pulsed and it's high intensity, like with deep brain stimulation or transcranial magnetic stimulation, they will respond in a different way. But with TDCS, we're talking about a low intensity, slow DC wash. That produces a low intensity, sustained neuronal polarization. And this, again, is not a question mark. It's something that my lab, other labs have demonstrated over decades. When you polarize neurons a little bit, you can change their function a lot. A little bit can go a long way when you're talking about a neuron, which is very nonlinear. For example, neurons fire action potentials. And to get to an action potential, their membrane potential needs to go up, 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 until you get to the threshold, and then it fires an action potential. Now, if a neuron is right near that threshold, which means it's gone up, up, but it hasn't made that final step up to generate an action potential, now we add direct current to the mix, and we produce that final little up, right? A tiny little dial up of the knob. Yeah, that makes a world of difference because we've gone from no action potential to a full action potential. So there's been a lot of work with TDCS that explains how a little influence or like a little push can go a long way.
Yeah, it's kind of like changing the sensitivity, or like the, like on a trigger on a pistol, like being able to move it a lot, like needing to pull a lot to shoot versus not. I heard that salt does the same thing for taste buds, like it kind of just amplifies everything.
That's a great analogy. It's changing the sensitivity. So now, if you don't have any input, and this is true for TDCS and for taste as well, if you don't eat anything, you're not going to taste anything. So it doesn't, salt maybe is a little different, but the basic idea is, it doesn't do much, or it doesn't do anything interesting on its own. What it does is it changes the tongue or the brain's response to something else. And so a lot of times when we think about TDCS, we think about changing the sensitivity, and then the question comes in, well, the sensitivity to what? Often, TDCS is combined with other forms of therapy, like behavioral therapy, right? Or physical therapy. So the therapies are let's call them the steak, and the TDCS is the salt. The therapies are what's primarily driving the benefit. It's primarily what's driving, what we call plasticity in the brain. And the TDCS is the salt, gives you more bang for the buck.
Yeah, definitely. That's a fascinating stuff. So you guys work on this, and how do you apply? I mean, you put it on, and you apply this electrical field, and then you have to do some kind of experiments, or like learning or something like this to amplify whatever message, or what's your guys' game plan?
So obviously it would depend on what you're using TDCS for. TDCS or these and related techniques. I should mention there's transcranial alternating current stimulation. There's a version of TDCS called high definition TDCS that we've developed. So there's a family of them. But in general, you have experiments where it is being applied on its own. Now, in those kind of experiments, you can ask questions like, does it help people focus? So they're doing something really dull. Does it help them pay attention on what they're doing? Or if you try a different flavor or mode of stimulation, can it help them relax? Can it help them transition to sleep? So that would be one kind of experiments. But then you have other kind of experiments where people, for example, are trying to learn something. And now you're applying it at the same time as they're trying to learn something to see if you can, they use this term accelerated learning, right? If you can make them learn faster, or learn to a level that they could not otherwise have gotten to with the same interval. And obviously everything I'm talking out here can be done with healthy subjects. There's a whole other range of experiments of how this is tested in when you're looking at clinical populations and you're looking at therapy. There, again, you're often combining TDCS with some form of therapy. So we just started a trial looking at home-based TDCS for depression. And in this case, there's sort of a guided meditation-based therapy that is combined with TDCS. So again, going back to the theme that in many cases, not all cases, but in many cases, it's being combined with something else, trying to create a synergy.
Yeah, that's really interesting. And then, so you do. Let's talk a little about your research. What you focus on there? What are kind of some breakthroughs that you've had there?
So my group works in sort of different domains. Some of those domains relate to just basic cellular mechanisms, right? Understanding at the cellular level what TDCS or other forms of neuromodulation do, because I believe that's really the basis for translation, right? You take these insights and then you use them to optimize technology for specific indications. But you can't do that if you don't at some core level understand the mechanisms at the cellular level. So one class of experiments, or one that we've been conducting over years, looked at how direct current stimulation could affect plasticity. So in animal models, there's synaptic plasticity. It's called neuroplasticity. This is thought to underline learning, though in cells you don't really have learning. You have this analog of learning, neuronal plasticity. So neurons changing their connectivity in one another in a lasting way, in a durable way. And this is thought to underlie how learning works. So what we can do is at the cellular level, we can sort of we can fake learning. We can use different techniques to sort of trigger the learning process. And now we can apply direct current stimulation on top of it. And what we found is that we can boost this learning. We can boost this plasticity. We cannot generate plasticity de novo. We can't create the learning. But if this learning, and I'm using learning, in this case in the animals, this plasticity is already ongoing, direct current stimulation is a very powerful booster. And what's also interesting is that if you repeat the learning, this is now in tissue again, so it's plasticity, right? You do the plasticity again and again and again, you'll eventually plateau. There's sort of a ceiling of how you can get. When you apply direct current stimulation, you get more out of the first round of learning, but also more out of the second round. And then, whereas you would normally plateau at a certain level without direct current stimulation, you plateau at a higher level with direct current stimulation. So we don't just accelerate the learning, we can actually get you to a place where you might not be able to get to otherwise. And again, I'm being very loosey-goosey here. Again, the experiments in animals are with plasticity, but also by relating that to learning, how this is a basis for applications like using TDCS to enhance neurorehabilitation becomes very clear. Neurorehabilitation, whether you're working with a therapist or a robot, is driving plasticity in the brain, right? You're trying to teach the brain new tricks. Now you apply TDCS on top of that, and you make the neurorehabilitation more effective.
That's fascinating. Yeah, I had heard about I think it was like an NPR interview or something like this, NPR story, and she was like shooting, enemies or something like this on a video game or whatever. And then she put it on and she was like it was like being in the zone kind of, right? Is that how you would describe it? And then what kind of percentage gains can you expect with this?
So what you just described is these immediate effects, these acute effects, right? Which I think can relate to plasticity, but don't necessarily depend on it. So you're putting on the TDCS, and it is very common for people to report an immediate change in how they feel or how they're thinking. And I know the article that you're talking about, I think she went to some sort of shooting range or something, and she performed a certain, it's an anecdote, but it's an interesting one. And then she put the TDCS on it, and I forget, she was like a perfect sniper, or whatever it was. But as anecdotal as that is, there are many labs that have run sort of controlled trials doing similar things, where people are doing something and they're applying TDCS and they're looking for an immediate change in performance or immediate change in report of how people think and how they feel. And for me, at some level, that's not surprising, right? I mean, the brain is electrical. Everything about how we think or how we feel is electrical. So it's not a shocker that when we apply electricity on top of this system, we can somehow modulate it. Now, again, that's kind of hand wavy, but that's sort of the starting point.
Okay. Yeah, it's fascinating stuff. And then you have the company Soterix Medical. What do you do there? And this is offshoot of some of your research, and then now you're able to apply it in industry. And I love this. I love this kind of crossing the chasm of bench to bedside, because it's just that it's not done often enough. So first of all, congratulations on being able to do that. But what do you do there, or what does the company do?
By the way, I agree with what you're saying, that it's not done often enough, and there's many reasons for that failure. And the people, obviously the people who are ultimately suffering for that failure are patients. And so there's a general issue with how we do translation. I started Soterix Medical, must have been about over a decade ago. At that time, it was very much a spinoff of my lab. It was motivated by an invention in our group of something called high definition transcranial direct current stimulation. Regular TDCS, as I've described it, uses two large sponge electrodes that are placed across the head. With high definition, we thought, well, instead of using these big sponges, why don't we use small electrodes that we called high definition electrodes? Instead of just using two, maybe we can use many, and maybe we can position them across the head and energize them in a very specific way in order to achieve targeted transcranial electrical stimulation. At the time, this was thought by many to be impossible. People said, well, the skull is quote unquote resistive, so you just can't be non-invasive electrical and focal. If you want to be focal with electricity, you got to drill a hole in the skull and put something in. or use something completely different like transcranial magnetic stimulation or ultrasound. So we showed, nope, it just requires a little bit of engineering, and you can be focal with electrical stimulation. That idea generated a lot of interest. And at that time, there was no, this was, by the way, started with a computational model, and we did some theory. And so at that time, there was no device that could actually do it to allow people to test whether this idea was true or not. So we started to design that device in my lab. There was a lot of interest in labs that wanted to obtain that device. And so it was very natural at that point to a company to actually produce this high definition transcranial direct current stimulation. Now, since then, Soterix has grown, and it continues to make that particular product, but it makes many new additional products. And it remains sometimes the case that sometimes something that we discover in the lab can be very quickly translated to the field, because Soterix Medical will be able to pick it up, productize it, produce it to FDA quality systems, address all the regulatory issues, and then provide it to colleagues who will then test it on for whatever questions that they want to answer. So there is a very, the lab is obviously separate from the company, but there's a pipeline that is very efficient. I also do work with many other companies, I should say, not just Soterix. The lab has a lot of resources that I think are attractive to many other small and large companies as well. And so we do have other avenues for translation separate from Soterix.
Is this talking about the consulting side of things as well?
Yeah, I mean, the agreements can take form of consulting. They can also take form of sort of just research contracts to the school. So again, we're very fortunate that we have some very unique and advanced facilities at the center I work at, and so even very large companies, right? Companies like GlaxoSmithKline or other sort of brand name companies that you'd hear about. Even these very large companies often lack certain resources. They need something very, very specialized. They have a pain point in their product development, and they will seek out specialized academic centers to help them out. And my lab is one of those academic centers. And so we end up doing quite a bit of contract work for industry.
Okay. How does that work? I mean, I haven't heard many guests talk about that, I guess. Is there something, I mean, is there a reason maybe it's not done very often, or maybe not talked about often, or I guess what are some challenges with this?
I don't know. I mean, there are, like with everything, there's some mechanics to everything, right? How are these contracts arranged? How is intellectual property protected, right? These companies are very sensitive about that kind of issue. And guess what? So are universities, right? So all the lawyers have their claws out. But, I would say that there is a way, and there are templates for how these contracts can be developed. And it certainly has helped that we've done this several times before, so that, my university has its templates, or when a company comes with its templates, we're familiar on what you basically need to accept, but what is fair to push back on. And I have found that it is very easy, in a couple cycles to come to terms that everyone sees as beneficial, and then to execute this work. I mean, it could be a little bit different than conventional academic work. For example, in conventional let's say NIH funded work, you're rushing to publish, you're rushing to share. While in the case of contract work, there may be either a short term or a long term block on the ability to share that work, which makes sense. The whole point is that these companies want to translate this technology to make it available to people. And we have to support them in that mission. It's why the work is so meaningful, right? It's why I love doing contract work for companies, because you're one step away from making it available. So there are things that we need to navigate and maybe kind of adjust our thinking a little bit. I think the fact that we do a lot of that kind of work makes it easy for companies to work with us, as opposed to maybe centers that don't. But in principle, it's such a synergistic situation. You know what I mean? It's great for me. It's great for the students. They're seeing cutting edge applications. They're working with industry. They're understanding what's important and also what's not as far as translation. And the companies are getting access to stuff that is really hard to cultivate in-house because it's so specialized.
Yeah, even something that's, seemingly simple, you're like, oh, okay, we can recreate that, to get it up and running and get it working, takes skills, takes, technicians, and it really is something that might take, a long time for a company to spin out, and it might be much more expensive and take much longer than it would if, under your careful hands. Yeah. So
this, by the way, includes, it could include access to just like you took equipment like, MRI. So we're very fortunate that we have a scanner right in our building. It's a research scanner. It could be, we can stimulate inside the scanner. Even very, very large companies, they're not, the biggest companies in the world, right, are not going to put one of these scanners on their facility. It's just something that's not done, for many logistical reasons. And the same thing goes for sort of human research in general, right? When you want to, whatever you're doing that involves human research, this is a very sensitive topic for many companies. And there's many reasons. Some companies are quite happy to sponsor that kind of work, but other companies prefer to let someone else manage that process for them.
Yeah, for sure. For sure. It makes a lot of sense and most of the time to do that, and a lot of times to do that. You also, I think you said co-founded and you help run NYC Neuromod. Do you want to talk a little bit about that? And then what have been some of the challenges in this last year as well?
So, I never really saw myself as like a conference manager person. This thing just something emerged organically. NYC Neuromodulation has now run, I think, about 5 times. The last time was an online version. The 2 times before that, we worked with other organizations. The North American Neuromodulation Society was partnered for one, and Neuromodulation the Science was partnered for another. I started the conference. It's called NYC Neuromodulation. So, often things are called what they are. The point of this conference, though it was an international meeting from the beginning, was to really put a spotlight on all the neuromodulation work that was going on in the New York City area. There's a massive concentration of neuromodulation work here, but maybe not as much of a community or not as much of recognition as in other centers like in Boston. So I wanted to bring that community together and put a spotlight on it while obviously also bringing the best from everywhere to this meeting. And, the first meeting was a success. And so you kind of want to do it again and again. Conferences can be very difficult and painful to put on, especially when, there isn't an organization behind it. So there isn't like a NYC Neuromodulation organization. This is really being put on by my university and my lab and myself, and also my colleagues who sort of volunteer their time. But the product is something that's beautiful, right? Or hallways packed with people exchanging ideas and just wonderful talks that you may not see anywhere else. And so it's such a joy to see that come together. Young people who are given their first opportunity to get on the stage, or not so young people, just seeing this as a conference where they can kind of share their latest and greatest to an audience that really wants to hear about it, and bringing industry there and having the right kind of interactions with industry. Often industry is like, there's like an exhibit hall and then there's like the talks, right? But it's this sort of artificial divide where there's a lot of research going on in industry, right? That industry wants to share and academics want to hear about it. So I've also tried to work on that aspect of it as well. And there's a lot of other stuff that a lot of other sort of public good that a conference can do. But I'll tell you, like, each one of these conferences, it's like the most tired I've ever been in my life at the end of it. Like, just a level of exhaustion from having to just be like so on top of it for the whole time leading up to it, to the thing. And then it's like you do it again. I guess it's like having kids. Like, you just forget. Something happens and then you forget until you have another one.
That's funny. And then last year it was all online, and you were talking about before we started recording that it had a very high attendance. And then what's the plans for this year as well?
So the last NYC Neuromodulation was online. Um, we always try to be very innovative in how we organize it. So we said, look, often there's a sense of like, there's like an acceptance committee and you have to like submit, you know what I mean? And who gets in and who doesn't. I said, look, self-organize. I said, anybody can run a session. All you need is a Zoom account. So submit it. Give me the details. And the conference almost acted like a hub, right? Where we were just sort of tabulating when the different Zoom sessions were and directing people to how they can register that and doing some level of quality control. But we didn't limit anybody's ability to present anything. So if you had a bunch of graduate students who wanted to share what they wanted to share, they could have a session. Now, I didn't know how many people would show up to each one. A lot did. We had, we had a professional Zoom account, but we were limited to several hundred per room, and the sessions were just filling up. I mean, there was a pre-registration process, so we saw these sessions were filling up. So now we're trying to add more sessions and boost the account, and we ended up having thousands of people attend, all organic, all free, 100% free. We didn't charge for anything. Companies could run sessions, by the way, as well. And I thought it was a great success. Many of the recordings were optional. The sessions could choose to record or not. The recorded sessions remain online, so people can Google NYC Neuromodulation and they can find the last event, and they can at least watch those talks that people chose to record. And again, I would love to recreate that experience again, or recreate another wonderful NYC Neuromodulation experience. Right now, obviously, everything has to be, you have to consider the current situation. So whether that's going to be online or hybrid or in person, I'm not sure, but I can see myself forgetting how hard it is and doing it again.
Yeah, but it sounds like it was a good success. I mean, a lot of people have complained about the online conferences. It's just not the same. But actually, I spoke and I attended at that one too, and I enjoyed it. Like, I thought it was really well done, and because there were so many more topics and rooms and everything like this, it just spanned much more than it would normally. So I thought that was good as well. I don't know. Maybe that's the future. This kind of open source, just a platform to speak on.
And I think it's so much about the culture. I have been to online conferences where I'm there and I'm like, I don't even know why I'm here. Like, the interface is so poor and detached, and I feel like even though I'm watching something live, I might as well be watching a recording, or even it is a recording. And I might as well watch something on YouTube at that point. And yet there's other conferences, where you feel like there's interaction and something's happening and you're seeing a live event and people are interacting in real time. And it is possible to create those kind of atmospheres as well. It's up to the people running the event, what kind of culture they want to create inside their event.
Mm-hmm. Yeah, definitely. Well, Marom, you seem to have done a lot of things and have a lot of experience in various fields. I mean, literally everything, like around neural engineering, and having a company, consulting, being academia, putting together conferences. Like, it just keeps going. So what are some big pieces of advice that you would say to other neural engineers, or what are they? I don't know. What are some tips that bits of wisdom that you can impart
to young neural engineers? You gotta love it. I feel like, I don't know if that's like universal, right? It doesn't really matter if you're gonna like open up a coffee shop or something else. But I love what I do. I feel very privileged that I have the opportunity to do what I do. And I feel like many of the people I work with who are the most successful love what they do. And part of that love is it can take a lot of work. And at least for me, there's been a lot of setbacks, a lot of no's, a lot of rejections. Like, that's par for the course. You know what I mean? I'm supporting a younger scientist now who's trying to get a faculty position. They've applied to like 40 positions. They've only gotten one interview, never mind a position. So it is a hard process. I feel like if you love it, that kind of gets you through it. It lets you can't enjoy all of it, but hopefully you can at least enjoy most of it. You know what I mean? And so maybe that's a start. And I think that if you're in a situation where you're finding that you're not loving what you do every day, that's not a reason to change your career. That's just a reason to maybe change how you go about engaging in that career. So there's many neural engineering is a very big tent, and there's many different people doing translation, communication, R&D, management. There's a lot of pieces into it, and so people can sort of try to find their niche. I mean, one other thing I see a lot is there's a lot of pressure to specialize. Right. I mean, when you're picking, like, I'm going to triple major as an undergraduate and do research in this topic. And then when I go do my PhD, it has to be like about like, the left CA1 region of the, it's like this, like have the hippocampus, like this sort of micro specialization. And I feel there's two sides to that. On the one hand, obviously we are specialists, and our value, right, as technologists, as engineers, as professionals, come from some level of like knowing something that other people don't, right? But there's another side to it, which you have to realize, like, you're probably not going to be working on the same thing in 10 years that you're working on now. So you're invasive now, in 10 years you're non-invasive. Your research now, in 10 years you're focusing on, like, I don't know, communication with a particular patient group, right, or something like that, or you're a sales rep, or you're doing something very different. And so I think that maybe should free people up. That if I'm saying, if you get a PhD or a job that's not exactly in the specialty that you want, but you're going to thrive, versus another position which is exactly what you want, but like your boss is going to be a horrible person and it's going to make you miserable, you're better off with that first option, right? Because you're playing that long game, and you're good, and as long as you're successful in that next stage, it will launch you into that future pivot. I don't know if I was clear about that, but I'm also saying you got to be good at something, right? And so you have to. Those are the. It's not even two sides of the same coin. It's like two coins. No,
I mean, if I did put it together, I would say, yeah, you have to play the long game and make long-term decisions. But in order to do that, you have to be enjoying it, because you can't be faking it, for the long term. You can't be like 10 years of like, oh, I hate this, but I secretly, I'm still going at it, something like that. So I don't know. I think those are the same thing.
But yeah. And in principle, it should we in neural engineering, neurotechnology, the pieces are there to enjoy it. We're studying the brain, like the most interesting organ. This is the frontier, right? Yeah, it's interesting to go to Mars, but there's another frontier, right? That's so much more exciting. That's right inside our heads. And as a result of that, we're creating the technology of science fiction, right? We're doing it, right? So it couldn't be more exciting. And we're helping people who are suffering. They're depressed and they're in pain. You know what I mean? And so we're helping them. And at the same time, with healthy populations, we're like expanding the limits of what it even means, I don't want too bombastic. We're expanding in some of the limits of what's possible, right? For healthy individuals as well. So as a field, it's so exciting. And so that we have that going for us, right? But it doesn't mean that everyone finds himself in a nurturing environment, or in an environment where they're being supported. And so you have to make that work as well. And you have to find a path for you that is that, like you said, like you kind of, if you're loving what you're doing, it's probably a good sign that you are on a good long-term trajectory.
Yeah, that's a really good sign. That's a really good point. Yeah. Yeah. Well, I think with that, we can maybe finish up. That was a great closing thought. If you're loving what you're doing, then that probably means you're on the right path. Very cool. Is there anything that we didn't talk about that you wanted to mention?
No, I really enjoyed this. I appreciate your questions, and I do like that we ended on that note.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.